In this video, we will discuss about glycosylation in cellular functions, and it also helps in maintaining homeostasis of the cells. So, glycosylation is one of the post-translation modification that occurs around 50% of the proteins that are being produced by mammalian cells. So, glycosylation mainly helps the protein in towards stability and folding. We have discussed during our initial videos of this channel that how the intracellular chaperones helps this proteins in the maintaining its stability and proper three-dimensional structures, which is essential for its regular functioning. And that thing, intracellular chaperones such as calnexin and calreticulums, works based on the initiation of the N-glycans in the endoplasmic reticulum. And other things that they affect is cellular signaling and communications. The glycosylation on the receptors also plays a role towards its proper activations. That is shown in this example that if the glycosylation is proper, then it can interact with its counter ligands, whereas if the glycosylation is not proper, it won't be able to it might not be able to interact with its receptors, or sometimes over interactions can also occur. And since there is a disruptions in the glycosylation profile in the cancer cells, and it has been found that those glycosylation profiles around the cancer cells prevents the immune systems to come and detect the cancer cells, and thereby helps the cancer cells towards immune evasions. And at the same times, because of the change in the glycosylation profile, it can also help in the cancer cell detections. So for examples, in the pancreatic cancer cells, one of the commonly accepted biomarkers is CA19-9, which is based on Levi's silyl structures, which is a glycan structures, one kind of glycan structures. And in this cancer adhesion and migrations, for this also we can in order to understand this thing, we can also consider the disruption in the glycosylation profile in the pancreatic cancer cells. There is a elongated and glycan structures that are found on the cell surface of these pancreatic tumor cells, and which will affect the cell-cell interactions and also helps in the migrations of the tumor cells. And another example is this cellular localization and trafficking. Because of the change in the glycosylation profiles, what it will does is that one of the example is that in the endoplasmic reticulum, there are more amount of unfolded proteins are formed because of the changes in the homeostasis within the cells, and thereby some of those proteins will be directed towards the lysosomes, and this will increase that pathways, that is ERAT pathways, endoplasmic reticulum associated degradations, and that will disrupt the homeostasis of the cells. So this is one of the examples how these cellular localizations and this trafficking will occur. And one of the positive side is that it will protect against the degradations. So some of the if the silations is occurs in a normal manners, then what it will does is it will prevent the proteolytic enzymes to come and degrade the proteins. Whereas if the proper silations or the proper three-dimensional structures are not formed, then it will be detected by the quality control mechanisms of the cells, and that will lead to proteolytic degradations using the proteolytic enzymes. This can occur either in the cytosols or in the lysosomes directly, and this is similar to what I have said in the cellular localizations and trafficking. And in these pathological changes that I have just mentioned during the start of this video, that there is a change in the glycan structures, and we have also discussed in our yesterday's video, that will lead to the immune evasions in this case that we have discussed. At the same time, these pathological changes will cause change in the glycan structures that can also be one of the therapeutic approach to detect the presence of tumor cells within the patients. And apart from CA19-9, there are some of the other biomarkers are also detected, which are also glycosylated. So we can combine those other biomarkers along with CA19-9 to detect the early detections of the pancreatic tumors within the patients. And this role in next major factors where it plays a role is in blood types and immunogenicity. We have known that a particular kind of individual can receive a particular kind of blood types only because of this immunogenicity. If the patient has like B positive blood, then it can receive the blood from B positive and one more universal candidate. And I'm not able to recall what's the universal candidate, because if we take the bloods from any other blood types, then they will have different glycan structures, and that will trigger the immunogenicity, and the immune system will degrade or reduce the half-life of the blood that has been induced. At the same time, it will also cause problems such as immune reaction, hyperactivate the immune systems. So these are some of the effects and some of the positives and negatives of this glycosylation in the cellular functions, and how they help in the homeostasis in the normal conditions. If you need to understand more about the role of glycosylation, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.